Imagine a patient who has spent, let's say 20 years trapped in severe, relentless, just daily depression, and every traditional medication has completely failed.
Right. A very real, very common scenario, sadly.
Yeah, exactly. Now, imagine that same patient undergoes a single, like maybe 6-hour intervention, and when doctors follow up 6 months later, that depression is still in complete remission.
It sounds like, science fiction. Or maybe something out of a mystical retreat.
It totally does. But today, we are going to show you exactly how this isn't magic at all. It is microscopic biological math. Welcome to this deep dive. Today, we're exploring a really brilliant piece from the Substack called SpiralSeekr, and it's titled The Role of the 5-HT2A Receptor in Psychedelic Pharmacology.
Yeah, it's a great read.
The goal here is to completely bypass the media hype and, those flashy headlines you constantly see about the psychedelic renaissance. We are going to look under the hood.
Because there's a lot of actual hard science there.
Right. We are tracing the journey of a molecule from the exact moment it hits a single cellular lock in your brain, all the way up to a total restructuring of human consciousness. Okay, let's unpack this. We're starting at the absolute ground floor, like the microscopic level.
Yeah, so the foundation of this entire biological cascade rests on one very specific, very tiny structure in the brain called the 5-HT2A receptor.
Okay.
This is what we call a G protein-coupled receptor. Now, its everyday evolutionary job actually has nothing to do with hallucinating.
Right, our brains didn't evolve just to, trip.
Exactly. Its natural function is simply to bind the neurotransmitter serotonin. That's what it's there for.
And these receptors aren't just scattered randomly, right? I mean, the Substack notes they are heavily concentrated in the neocortex, but what does that actually mean for the architecture of the brain?
Well, it means they are positioned right in the command center. Specifically, they sit on the apical dendrites of layer V pyramidal neurons.
Layer V pyramidal neurons? That sounds intense.
It does. But if you visualize these pyramidal neurons as like ancient towering trees in a forest, the apical dendrites are the long trunks reaching all the way up to the very surface of the cortex. They are essentially major communication highways.
Oh, wow. Okay, I like that visual.
Yeah, and you also find these receptors clustered in other key regions like the colostrum, the thalamus, and the basal ganglia. And these are all areas responsible for filtering sensory information and really governing consciousness itself.
So on a normal Tuesday, when there are no psychedelics anywhere in the system, what is the 5-HT2A receptor actually doing up in those trees?
It acts as an excitatory trigger. So when your natural serotonin binds to it, the receptor modulates the release of other neurotransmitters. And the main one is glutamate.
Glutamate.
Right, which is essentially the brain's primary excitatory chemical.
So it's basically a master volume knob for the brain's excitatory signals.
It's really a way to put it.
You turn that 5-HT2A knob and you flood the area with glutamate. You get more activity, more chatter between those big cortical trees. But how do we know this specific knob is the trigger for a psychedelic trip? Like, couldn't a complex substance like LSD be doing a dozen other things across the brain simultaneously?
That is a crucial question, and it was actually answered by a foundational paper published way back in 1984 by a researcher named Richard Glennon and his colleagues.
1984. So we've known this for a while.
We have. They looked at classic psychedelics, so LSD, DMT, mescaline, and psilocin, which is the active compound your body converts psilocybin into.
Right.
And Glennon's team discovered a direct, undeniable mathematical correlation. If you want to know how powerful the hallucinogenic effect of a drug is going to be, you really only have to look at how tightly it binds to that single 5-HT2A receptor.
Wait, really? Just that one receptor dictates the entire behavioral potency?
Almost entirely. I mean, the stronger the chemical grip on 5-HT2A, the more potent the reality-altering effect. But what's fascinating here is the ultimate definitive proof of this mechanism.
Which is?
Well, in human trials, researchers can administer a compound called ketanserin. And ketanserin is a 5-HT2A antagonist.
Meaning it occupies the receptor, but it doesn't activate it. Like, I don't know, jamming a broken key into a lock so no other key can get inside.
That is a perfect way to visualize it, yes. So if you give a human subject ketanserin, their 5-HT2A receptors are entirely blocked.
Okay.
If you then give them a massive, highly potent dose of a psychedelic,
nothing happens.
Nothing, absolutely nothing. The subjective experience, the visual hallucinations, the emotional shifts, they are completely neutralized.
Wait, hold on. You're telling me you can give someone a heavy dose of LSD, but if you've taped over that one specific microscopic keyhole, the drug does absolutely nothing.
Nothing at all. The profound alteration of space, time, ego, It is entirely shut off.
That is wild.
It is. It grounds an experience that has been described as deeply mystical for thousands of years in hard, undeniable neurochemistry.
That is staggering. But, it also creates a glaring paradox. And if you're listening to this right now, you're probably wondering the exact same thing I am.
Which is?
If these psychedelic molecules are just binding to the exact same receptor that my natural serotonin uses every single day, why aren't I tripping right now? Like,
Right.
why doesn't a surge of normal serotonin send me into a hallucination?
That is the million-dollar pharmacological question right there.
Mm.
To understand why serotonin keeps you grounded while psilocybin sends you to outer space, we have to zoom in even closer.
Closer than the receptor.
Yeah, we have to look at what happens inside the cell after the receptor is activated.
Because binding to the outside of the cell is just ringing the doorbell. It's who answers the door that actually matters.
Exactly.
Yeah.
When any molecule binds to the 5-HT2A receptor, it causes a conformational change. The receptor physically twists and changes its shape. And this twisting activates proteins resting just inside the cell membrane, specifically the G-alpha-Q/11 proteins.
Okay.
Now, historically, neuroscientists mapped out a very standard cascade of chemical messengers that get released when this happens.
And the Substack goes into really deep detail on this. It brings up a pretty dense alphabet soup of enzymes and messengers.
It does, yeah. It mentions phospholipase C, hydrolyzing PIP2, which splits into DEG and IP3, and then...
Let me stop you there, actually, because I want to make sure we don't lose the forest for the trees here.
Fair enough. It's a lot of acronyms.
Right. Instead of memorizing alphabet soup, let's talk about the function. Like, what is all of this machinery actually doing?
Okay, think of the receptor as a complex fire alarm pull station on the wall of the cell.
Okay, a fire alarm.
Right. When your natural serotonin pulls the handle, it twists the mechanism in a very standard way, and it rings a bell inside the cell. It's a balanced, predictable, normal response. But when a psychedelic binds to that same handle, remember, it's a completely different molecular shape. So it twists the pull station in a bizarre, asymmetrical way.
Ah, so it fixed the lock, but it turns it at a weird angle.
Yes. And because of that weird angle, it doesn't just ring the bell, it triggers the cell's internal sprinkler system instead.
Oh, wow.
Yeah, it activates completely different secondary pathways that serotonin largely ignores. And in pharmacology, this secret backdoor concept is called functional selectivity, or biased agonism.
Biased agonism? So the psychedelic is like a biased key?
Exactly. Our endogenous serotonin is unbiased.
Right.
It activates all the cell's pathways equally. But psychedelics are biased agonists.
Okay.
They preferentially trigger distinct signaling cascades, like the beta-arrestin 2 pathway.
Yeah.
Or they force the 5-HT2A receptor to physically pair up with neighboring receptors, forming what we call heteromers, like the 5-HT2A and MGLR2 complex.
This makes so much sense. Like, it explains the paradox perfectly. The psychedelic isn't just mimicking serotonin at all. It's hijacking the hardware to run a completely different software program inside the neuron.
That's a great analogy.
But, that brings us to the next massive hurdle. We've got a biased lockpick, and we've got a weird chemical cascade inside a single cell. But one cell doesn't make a conscious human.
No, it definitely doesn't.
So how does a change in these isolated cellular sprinklers ripple outward to fundamentally alter the entire architecture of the brain?
Right. To see that ripple effect, we have to move from the microscopic to the macroscopic. We have to look at brain network dynamics.
Okay, zooming back out.
Yeah. When millions of these 5-HT2A receptors are being stimulated in this biased way all across the cortex, the brain's massive established networks begin to destabilize.
And researchers don't have to guess about this anymore, do they? The article highlights functional MRI research, specifically pointing to landmark studies from Carhart-Harris and colleagues in 2012 and 2016.
Yeah, the fMRI data is really where the mystical becomes visible. When researchers put subjects in a scanner and administer a psychedelic, they watch the brain's normal functional networks literally dissolve.
Just broke apart.
Yep. And the most critical system impacted is the default mode network, or the DMN.
Now, the DMN is such a wild concept, even without psychedelics in the mix.
It really is.
It is essentially the brain's background processing system, right? Like, it's most active when we aren't doing any specific external task.
Exactly.
When you are daydreaming, when you are worrying about a conversation you had 3 years ago, or when you are just thinking about your identity. That is the DMN running the show. It is the neurological neighborhood where the ego actually lives.
That's spot on. And acutely, under the influence of a psychedelic, the integrity of that default mode network collapses.
Wow.
The blood flow drops, the synchrony drops, it goes offline.
Yeah.
But here's the thing. The brain doesn't just shut down. As the DMN dissolves, global functional connectivity absolutely explodes.
Here's where it gets really interesting. Think of the brain's networks as a massive, deeply rigid corporate office.
Okay, I like where this is going.
So the default mode network is the ultra-strict boss. It keeps everyone in their designated cubicles, it maintains the hierarchy, and it strictly dictates who is allowed to talk to whom. The visual cortex stays in its lane, the memory centers stay in theirs.
Very rigid.
Right. And when you introduce a psychedelic, it is the equivalent of sending that strict boss on a sudden, unannounced vacation.
That is a phenomenal way to frame it. Without the DMN constantly suppressing communication, the brain enters a state called brain entropy.
Entropy.
Yeah, and in thermodynamics, entropy means chaos or disorder. But in neurobiology, it means high potential and high flexibility.
So the boss is gone, and suddenly the accounting department is throwing a massive party with the graphic design team.
Exactly. Parts of the brain that haven't spoken since childhood are suddenly sharing information directly.
Yeah.
You have intensely novel patterns of communication crossing over the brain's hemispheres and lobes.
And that's what causes the trip.
Precisely. This temporary dismantling of the usual filters is what causes the subjective feeling of ego dissolution and those profound, deeply unusual insights people always report.
Hold on. A temporary corporate party in the brain is one thing. Eventually, the drug metabolizes.
Of course.
The boss comes back from vacation. The DMN boots back up. So if this state of brain entropy only lasts for, say, 6 hours, how does a temporary software reboot lead to the permanent behavioral changes we talked about at the very beginning of the show?
Well, because it isn't just a software reboot. The drug also forces a hardware upgrade. And this brings us to the concept of neuroplasticity.
The physical rewiring of the brain. The Substack digs into this, referencing studies that show sustained 5-HT2A activation actually causes structural changes.
It does. A major 2018 study by Lai and colleagues demonstrated that psychedelics promote 3 distinct types of physical growth in the brain.
Okay, what are they?
First is neurogenesis, which is the growth of new neurites. These are the branching projections coming off the main body of the neuron.
Branches, got it.
Second is spinogenesis, which increases the density of dendritic spines. Those are the tiny little knobs on those branches where connections are actually made.
Okay.
And third is synaptogenesis, which is the actual formation of new functional synapses between neurons that weren't connected before.
I want to make sure I'm fully grasping the scale of this. Like, we aren't just talking about a temporary chemical wash. We are talking about the brain physically sprouting new anatomical structures.
If we connect this to the bigger picture, this physical remodeling is the holy grail of psychiatric medicine. The psychedelic triggers the upregulation of a protein called BDNF. That's brain-derived neurotrophic factor.
Okay.
You can think of BDNF as high-powered fertilizer for the brain. And simultaneously, it activates the mTOR signaling pathway, which is essentially flipping the power switch on the cellular factory that builds these new proteins.
So the drug creates an environment where the brain is hypersensitive and aggressively building new physical bridges.
Yes. And this rapid, profound neuroplasticity is the biological bridge between a fleeting 6-hour hallucinogenic trip and a mental health benefit that can endure for months or even years.
Wow.
It is the core reason these substances are being investigated for treatment-resistant depression, PTSD, and severe addiction.
Which brings us beautifully out of the isolated laboratory and into the clinical reality of human suffering. How does all of this receptor binding, the entropy, and the BDNF fertilizer actually play out for a patient sitting in a therapist's office?
The clinical data from the last decade has completely upended traditional psychiatry. The article highlights a landmark 2016 study led by Roland Griffiths at Johns Hopkins.
Right.
They worked with patients facing life-threatening cancer diagnoses who were suffering from just debilitating end-of-life depression and anxiety.
Which, frankly, is the most understandable and difficult to treat form of depression imaginable. I mean, you can't just tell someone they shouldn't be anxious about terminal cancer.
No, you can't. It is incredibly deeply rooted. Yet, researchers found that a single high dose of psilocybin produced a massive rapid decrease in both depression and anxiety.
Just one dose.
One dose. And even more astonishing, when they followed up with those patients 6 months later, the symptom remission was still highly significant. Half a year of relief.
And the success isn't just limited to palliative care, is it? The Substack also covers a massive Phase 2 trial from Carhart-Harris in 2021.
Yes, that was a big one.
And this one was huge because it didn't just test psilocybin against a placebo, it pitted it directly against a standard daily SSRI medication, escitalopram, for treating major depressive disorder.
That was an absolute watershed moment, comparing the old paradigm of daily pharmaceutical symptom management against the new paradigm of acute psychedelic intervention.
And what happened?
The trials showed that a few sessions of psilocybin therapy were statistically non-inferior to taking a daily SSRI for reducing core depression scores.
But the nuance there is what really caught my eye, because the psychedelic actually outperformed the daily pill on secondary measures.
It did, yeah.
SSRIs are, they're notorious for blunting emotion. They raise the floor so you don't feel as sad, but they lower the ceiling so you can't feel deep joy either.
Right, emotional blunting.
But the psilocybin seemed to actually restore emotional responsiveness and psychological well-being. It didn't blunt the pain, it somehow fixed the underlying processing.
Which leads us to the ultimate question. How? How do we unify the biology of growing new dendritic spines with the psychology of healing trauma?
Right.
To answer this, researchers proposed the synergistic model, led by Davis and colleagues in 2020.
And this model completely reframes how we look at mental illness. It suggests that conditions like depression, severe anxiety, and addiction aren't just simple chemical imbalances.
No.
They are ruminative disorders. They are driven by heavily reinforced, highly rigid cognitive patterns. The needle of the mind is stuck in a deep negative groove, governed by an overactive default mode network.
Exactly. The brain is trapped in a rigid hierarchy. So bringing everything we've learned together, the psychedelic molecule hits the 5-HT2A receptor. The bias cascade suppresses the DMN. The rigid hierarchy is dissolved. The brain enters high entropy.
Right.
And simultaneously, the BDNF fertilizer floods the system, opening a critical window of neuroplasticity. The brain is suddenly highly malleable.
So what does this all mean? The way I see it, traditional daily pills are like putting a cast on a broken arm and just leaving it there forever to manage the pain.
Okay.
But this psychedelic model is more like a surgical intervention. You heat up the metal, that is the biological destabilization, and the brain entropy. You get it glowing red and malleable.
I love this.
Then you reshape it into something healthier. And finally, you let it cool and set into its new, upgraded physical form.
That analogy hits the nail on the head. But the most critical part of that process is the reshaping. The biological chemical cascade creates the hot metal. But the subjective, deeply meaningful experience of the trip, the psychological insights, that is the hammer that shapes the new connections.
And that explains why the clinical trials always pair the drug administration with intensive psychotherapy. You don't just hand a patient a pill and send them home to watch TV.
Never. The source explicitly points out that patients consistently attribute a high degree of personal meaning to their psychedelic state. The therapist is there to help the patient take those profound, highly emotional insights generated during the entropy phase and use them to actively forge new, healthier neural pathways while the brain is in that hyperplastic, malleable state. Biology and psychology are completely intertwined here.
It really is just a breathtaking sequence of events. Just to recap the sheer scale of what we've covered today.
Let's hear it.
We started with a tiny molecule acting as a biased, asymmetrical lockpick on a single 5-HT2A receptor. That triggered a bizarre internal cellular fire alarm. That alarm rippled outward to completely suppress the brain's rigid corporate boss, the default mode network, plunging the mind into a state of highly connected entropy. While the ego was dissolved, the brain's hardware literally sprouted new branches. And all of that physical remodeling allowed a suffering patient to reshape their trauma and find long-lasting relief from a rigid ruminative disorder.
It connects the most microscopic chemistry we can measure directly to the macroscopic reality of human suffering, healing, and consciousness. But this raises an important question, one that goes far beyond pharmacology and touches on the very core of human philosophy.
Oh, I love a good existential pivot. Lay it on me.
Think about the default mode network.
Yeah.
If our rigid sense of self, our personal ego, our daily anxieties, and our very perception of reality are entirely maintained by the continuous mechanical operation of this one specific brain network, and if all of that identity can be completely dissolved and fundamentally restructured simply because a different molecule flipped a microscopic switch on a single receptor, what does that say about the fragility and the purely chemical nature of what we call our true selves?
Wow. If the self is just a biological holding pattern that can be paused by a biased key, who is the one experiencing the pause.
Exactly.
That is a remarkably profound thought to sit with. Well, thank you for joining us on this deep dive into the pharmacology of the mind. Until next time, keep exploring and keep questioning the architecture of your own reality.